Well, I am delighted to welcome all of you to this afternoon's seminar. I'm David Relman, those we haven't met, and it's my wonderful, great pleasure to introduce my friend and colleague, Drew Endy, who is the Martin Family Fellow in undergraduate education and an associate professor of bioengineering here at Stanford, and a senior fellow by courtesy at the Hoover Institution. I think most of you know Drew, or know of Drew, and I don't want to belabor an introduction, but there were a few things I just wanted to make sure that everyone heard and had a chance to think about. Drew has a number of very important, interesting parts to his history, which have resulted in almost a unique kind of perspective and contributions that I hugely admire and respect for a number of reasons. But one of which is the fact that all of this has led Drew to express a set of visions for what you might view as the long-term welfare of our species. And this is the kind of aspiration and goal that many of us don't often think about or articulate, but Drew does so, and he does so in a very compelling way. Just a few comments about Drew's accomplishments. He is first a bioengineer in practice, an experimentalist. He is interested in building and strengthening the foundations and expanding the frontiers of synthetic biology. One example, a paper that he published this July in Nature Communications, describes the use of modeling approaches and experimental science to understand how the rates of protein synthesis might be predicted from a more precise exploration of quantities of the key components of the protein synthesis machinery in a cell. And of course, the goal is to be able to predict from the abundances and composition of these components and pieces protein synthesis rates, which is one of these fundamental features of what we might need to know towards the path of building a synthetic cell. And as a sort of a way of generalizing from this example of work that Drew pursues, it suggests to me anyway that what Drew is really seeking to do is optimize paths towards a more predictably productive future, in a more general sense. From a policy point of view, Drew has made a number of important contributions in the way of service and participation on a number of important and influential groups and committees and organizations, including the National Science Advisory Board for Biosecurity, in the past, the Committee on Science, Technology, and Law, the National Academies, the International Union for the Conservation of Nature's Synthetic Biology Task Force, and the WHO's Advisory Committee on Variola virus, smallpox virus research. He is currently doing many of these sorts of things, but in particular is the director of the iGEM Foundation. And I think you'll hear more from him perhaps about what iGEM has contributed and what it teaches us. And then finally, and fortunately for me, Drew is currently a colleague on a task force of the Defense Science Board in exploring how emerging biotechnologies might enhance a more successful pursuit of mission by the Department of Defense. Drew has been very active here at Stanford in teaching and in undergraduate admissions. In fact, he's the chair of the Committee on Undergraduate Admissions and Financial Aid at the present time. Drew likes to end his introductions with a few words that to him sort of embody the person who's about to speak. And so I was trying to think of a few words. It's hard to come up with just a few, but I think at the top of my list would be inspiring, provocative, incisive. I found, you may have read, there was a New York Times article that talked about Drew and being Drew. Somebody was quoted as saying that every time they heard him speak and talked to him, they felt as though their IQ had dropped a bit, only because it just seemed in comparison that, thoughts weren't happening at the same speed and level. But Drew really is a wonderful, engaging colleague. He's a community builder and a great communicator, as you either know or will soon see. He's going to speak today about policy failures and opportunities underlying 21st century biostrategies.
Thank you very much. Thank you, David. Thanks everyone for being here. I'll try my best. Going out on a couple limbs and welcome frank comments, upgrades, your best and honest thoughts. I wanted to start by going back in time and risk a little bit, a photograph from the 1930s in France. These are soldiers serving in the Maginot Line, a series of fortifications, and worried about apparently vitamin D deficiency. And so in that context, there's a UV illuminator and a person with a watch and trying to mitigate that potential deficit. A very rational set of actions in a historical context that was very, very sophisticated, but ultimately didn't secure sufficiently France. And so I've been thinking a lot about this book by Marc Bloch called Strange Defeat, which Marc writes as a Frenchman in 1940 and is struggling to understand what happened in France. And as a historian, practices his craft and goes back in time to 1920s and tries to make sense of 1920s to 1940. And so I just want to quote from the book. Bloch writes, "I want to consider the preparations that were made before the war broke out. If we were short of tanks, airplanes, and tractors, it was mainly because we had put our not inexhaustible supplies of money and labor into concrete. We had been taught to put our whole trust in the Maginot." Elsewhere in the book, he writes, "The generation to which I belong has a bad conscience. It's true we emerged from the last war, World War I, and the flu pandemic, desperately tired. We were only too anxious to get back to our proper employments. In today's parlance, let's get back to normal. That is our excuse," he writes, "but I've long ceased to believe it can wash us clean of guilt." Marc joins the French resistance, is captured and tortured and killed by the Nazis in 1942. His manuscript survives and it's published in 1946. I just want to acknowledge and honor that. This book keeps me up. I'd recommend it to anyone. What were the technical changes that were unfolding in the 1920s and 1930s that made it hard to make strategic decisions about how to invest the public treasure to secure the future? The internal combustion engine, radio, and a few other things. And with that as backdrop, we're now 100 years later in the 2020s. And what are the things changing? And are we acknowledging them well enough to secure our future? Or is one of us going to have to write the second version of this book in 2040? All right, so that's just as backdrop. What I thought I would do is give a read-in to my view of where biotechnology is going with evidence, and then get to, and here's the risky part, or the more risky part, things we might want to consider changing with respect to policy and strategy. And then we can talk about it. All right. So I wanted to put biotechnology in a context. It's both emerging and established. And so I think of biotechnology in 3 chapters. Chapter 1 is domestication and breeding. This is thought to be a very advanced R&D facility in South America, where the terrace levels control irrigation and temperature, and position controls sunlight. And in one spot, you can optimize crops for different regions throughout the Andes. And the crop of interest is the potato. Right? And so this is domestication and breeding in a high throughput centralized R&D facility. Best we can understand what that site was about. So this is generation biotech, still practiced today. Generation 2 starts 40 plus years ago, matures and matures and matures, most famously and recently as CRISPR. This is the gene editing generation of biotech, from recombinant DNA to Cas9 CRISPR systems, and the Nobel laureates associated with that powerful, most recent technology. The promulgation of CRISPR, in my view, is both because it's amazingly useful and accessible, but also it's so popularly promulgated because it fits within a governance framework that was established starting in 1975 for editing. It's still editing starting from existing materials, right? So it's new in important ways, but it's not totally new, this CRISPR gene editing era. I'm going to argue that synthesis is a third emerging still area of biotechnology. And synthesis involves putting things together. Here's an example of that. These bottles are bottles of the bases of DNA. So from right to left, A, T, G, and C. These bases are not derived from natural living organisms. They're made from scratch, starting from sugarcane and synthetic chemistry. And you can think of these as ink cartridges that you put into a printer, a synthesizer for DNA. And it's not, a musical keyboard would have 88 keys. This will have 4 keys. And depending on how you play the keys, A, T, C, G, the atoms will be organized due to dispensing of molecules to make your DNA from scratch, right? And you're not editing an existing piece of DNA, you're compiling a piece of DNA at the atomic level. It's a 1D printer, in other words. But it's a 1D printer that programs objects that make 3- and 4-dimensional objects, right? The ribosome in developmental biology. So why does this matter? If I want to make this claim that this is qualitatively new, I'll give you one example to start with, in the form of a question. How did SARS-CoV-2 first get to Switzerland during the beginning of the pandemic? And from the publicly available information, you can track the first case that's recorded. It comes in over the Italian border, hence the photo on the top left, and it's recorded on the 25th of February, 2020. That's not how the virus first got to Switzerland. What happened earlier was a researcher in China sequenced the genome of the virus, uploaded that sequence information to the internet, and the internet the next day was accessed and it was downloaded as information in the Swiss capital. Therefrom, the researcher ordered the DNA fragments encoding the genome of the virus for synthesis, got that assembled, in a test tube, read that out to make an RNA copy because it's an RNA virus, transfected that into cells in a laboratory, and was able to recover live infectious virus. And they did all of that by the 13th of February of 2020, i.e. 12 days before the first case is reported. Now, it could be that there were other infected people in Switzerland before. I'm sure there were, but I'm just going off the data I can read. So DNA reading plus the internet plus DNA writing moved this isn't the source of the pandemic in Switzerland, but it's how the virus first arrived. And so I call this the bionet, right? We're moving biological information around on the internet and compiling and growing. So that's what I mean by this is just qualitatively different than the domestication and breeding regime or the editing regime that also underlies biotechnology. And so how do we govern this sort of thing? This morning at 3:35 a.m., I got an email from a British company bragging about they just put their new DNA synthesizer in a lab at Imperial College. And I hadn't been tracking this company. The work that was done in Switzerland relied on a centralized gene construction company. So it's you send in your information to a company and like, make me this DNA, and they mail you it back. But this email this morning is a very different technology where it's desktop, right? And so you can see that next-gen gene synthesis, we are developing technology that will put a gene foundry in every lab, right? That's this morning. I haven't tried this platform exactly, so I can't represent it more than an advert. But I can talk about where gene synthesis technology is going, right? And the future generations of gene synthesis and DNA synthesis. Right now, DNA synthesis is mostly chemistry and physical hardware and plumbing. And the chemistry was perfected more or less by 1980. But the next generation of DNA synthesis is using enzymes and specialty chemicals. And then you can see a third generation, which uses enzymes and native chemicals that you can get in cytoplasm, meaning from biology. And then an ultimate form of a DNA synthesizer, there's one PhD student in the lab starting work on it, is an enzyme, a genetically encoded DNA synthesizer that you would control with light, 2 wavelengths of light to set the base and a third to say add or don't add. Now that's a fantasy right now, but if the PhD students are starting to work on it at Stanford, maybe somebody will figure it out by 2040. Right, so it's like the DNA synthesizers that we have today are these central facilities. They're going back to the tabletop, and then ultimately they might be genetically encoded themselves. Okay, so synthesis, putting things together. Obviously, at a macroscopic level, Biology is incredible at synthesis. This is a suspension bridge grown in place from the roots of a rubber plant, fixes carbon as it gets stronger. It's synthesis at the macro scale. I put this in here to remind us that if we can unlock biology as a general purpose technology and see it responsibly deployed, what the big prize is planetary scale flourishing. There's 90 terawatts of energy going in through photosynthesis for a civilization that runs on 20 terawatts, right? There's plenty of manufacturing capacity on the planet in a way that's compatible with life if we can figure out how to unlock it. So that's the big positive prize. I don't work at this scale, although I was a civil engineer a long time ago, so I love suspension bridges. I work at this scale, the molecular to cellular scale. This is a wonderful watercolor by David Goodsell of E. coli, the inside of E. coli and the flagella motor in green and so on, sticking out to the left. I'd love to be able to build something like this, and we're not able to do it. And I'm grateful you mentioned the most recent paper out of the lab, but let me show the most recent PhD thesis out of the lab that we don't even have up on bioRxiv yet, but it is in the Stanford Library. So this took us about 8 years to get to. This is a fully constructed synthetic object. We think of it as a synthetic egg. And what you're looking at up on the left is a electron microscope tilt series image taken off a cryo-EM rig that Hua-Chu up at SLAC operates. And you're just slicing through it and then tilting and false coloring. This object's about 500 nanometers across. It's got a lipid bilayer that we make by mixing up chemicals. And then inside it are purified molecules established by researchers in Japan 2 decades ago, the PURE system, that collectively are capable of expressing any DNA you add to the mixture that has the right start signals, right? And so by learning to put these things together and envelop them, in this case, we're showing that we can make a new RNA polymerase that we like better for purposes of cell building. The puzzle we're working on next is to figure out what DNA to add to this object to get it to grow and divide. And we've figured out how to debug that puzzle and established what this object can't make that's already in it. And so now we're just doing the patching reactions. If we can solve all of that, then we will have constructed life from scratch. That's not something that is now a graduate student project, in my experience. This is a very significant and challenging professional engineering project that ideally would be pursued at a national lab. But honestly, we don't have national labs that work on biology in this way, right? This is about $100 million of research, we estimate, to get this thing growing and dividing. But if we could pull that off, then we would be able to operate life at the cellular scale in a way that, from a naive engineer's perspective, we understand it. And to the extent that cells are the fundamental unit of life, that's a big deal. I think about it through metaphor this way. We exist in the constraint of a gravity well, and we use rockets to get up out of the Earth's gravity well and do interesting things, gain the elevation. I would say the Earth also has a life well. The life on Earth is constrained due to physics in different ways, from the lineage that comes before, and because life on Earth, in order to maintain lineage, has to be able to reproduce, a second constraint, and in a changing environment has to be able to evolve, right? So these 3 constraints of what came before reproduction and evolvability limit the life down in the Earth's life well. And so you can see the evidence of that. My maternal great-grandfather from Washington County, Pennsylvania, with the bow tie, apparently played the fiddle. And we have some patterns that are similar morphogenically, right? And I love the Hardy poem, I am the family face, flesh perishes, I live on, carrying trait and trace through time to times anon, and leaping from place to place over oblivion, right? That's the constraint of lineage and all its beauty, right? And all its wondrous pattern, but it's a constraint, right? And so if we can construct life from scratch, starting with simple living systems, we're perched up on the lip of the Earth's life well. It's not like the deep vacuum of space, rather it's just compile back down to whatever life could be, right? It's much more interesting in my view, and space is pretty interesting. Now, since we're not there, I thought I'd calibrate back to now and show you what synthetic biologists are accomplishing today and how they're translating it. So this is a cartoon schematic of brewer's yeast, and you see all the metabolism and biochemistry here. All of it's bioengineered, and this is work done by my wife, Christina Smolke, so I'm hopelessly biased, but it's the best example I can bring into the room and show you how it translates. And so in this case, brewer's yeast normally would take sugar and make ethanol and carbon dioxide. Instead, it's making a plant natural product called scopolamine. Scopolamine you could find in a motion sickness patch and is sometimes, I think, used for treating Parkinson's to a degree. And so they can figure out how to take something that normally be grown through farming and extracted and instead make it by a brewing process. This sort of thing demonstrates that the complexity of natural products, chemicals that are found in nature can be reconstituted basically at the limit of what we see inside yeast, right? So suddenly something you thought of as what's good for bread and beer and wine, now imagine it can make sophisticated chemicals at the limits of known complexity. Right? One thing to point out, Christina's PhD advisor, Jay Kiesling on the left, pioneered this work a decade ago. And the example on the left is making artemisinin by brewing for treating malaria. That involved 3 enzymes in yeast that were not found in yeast to make that product. And you see all the authors on the paper on the left, lots of people, about 100 postdoc years of labor. The work I was representing, just when you look at the academic paper, has 2 authors on it. Prashant, the PhD student, and Christina, who wasn't in the lab at all, for sure, right? And it's a 30 enzyme pathway, and Prashant did it in about a year. And Prashant might be the best person in the world at this type of stuff. He's amazing, but still, that's an incredible amount of work, right? And so something has changed in terms of we've gotten fundamentally better at composing biomolecules into biosystems. I don't want to go into all the weeds of that, but there's been significant fundamental progress underlying this. Okay, but then so what? Right, so an academic demonstration of making a little chemical isn't going to be impactful right away. Christina's left Stanford, she runs a company, and last month they reported that they've taken examples of these yeast strains, scaled them up in 120,000 liter fermenters at very high yields or titers of the chemicals, and it's working. And what's really interesting is before they did that, nobody thought it would work. And now that they've done it, it's like, oh, that's normal. It's very weird to see that transition. But to put this in context, 120,000 liter fermenter that runs for 4 days, and then the purification process is another week, so 2 weeks in a fermenter-like system, makes as much medicine as 10 square miles of poppies growing for 2 years. Right? So that's the physics transition. And what does a 100,000 liter fermenter look like? According to Alibaba, you can buy them for $50,000. I haven't tried doing this, right? But they're these just big steel tanks, and you have to be really good at operating them, so it's not trivial, but it's really an interesting transition. I went back and found a talk I gave in 2019, because for our purposes, it's one thing to think about medicines, but what are some of the other things maybe you could brew? And so here's an example I found from a talk I gave in 2019. A call from the Office of Naval Research to advance synthetic biology methods for making bioenergetic materials, propellants. All right, it's like, oh. Other things that are happening. Phil Ross got his Master of Fine Arts at Stanford, started a company called MycoWorks to use wood fungus to grow things. These are things that normally degrade trees that fall over in the forest. But if you feed these organisms with sawdust, they can grow very quickly. And the product currently going to market is, imagine a big lasagna tray of sawdust, and you mix in the wood fungus and it turns into a sponge. And then the top of the sponge differentiates into a mushroom skin that you can peel off and put through a tanning process. So now you have a surrogate for leather. Phil's company just opened up their first 100,000 square foot factory in Union County, South Carolina, next to the forest there. And that should make about 3 million square feet a year of leather surrogate. Back into the world of fiction, in 2018, the Semiconductor Research Corporation put out a synthetic biology roadmap. And they said, if we did $5 billion a year of research between now and 20 years from now, i.e. 2038, what could we get to? And it's written in chapter 5 here, biological pathways for semiconductor fabrication and integration. So as a complement to top-down lithography and classical manufacturing processes for semiconductors, we're going to use biology and its ability to do program pattern formation at the atomic to molecular scale and create scaffolds that guide the production of abiotic hardware. So we're going to use biology to build classical computers. Total fantasy today, given where the technology is, but I had nothing to do with this roadmap. I look at it, it's pretty good. It's not funded. The cost of funding the roadmap is about one chip fab in today's dollars that we're building to try and reshore. Right, so, I like this because of the impossibility of it. If you look at the history of electronics, it was World War II and the Cold War that caused the public treasure to flow to build out the engineering foundations of electronics, like really getting that solid. We've never seen that capital flow into the foundations of bioengineering. So I like this idea of bootstrapping biotech for technology as a way of hardening the engineering underbelly of bioengineering. Because otherwise in bioengineering, we mostly just get money for working on diseases, very applied work, right? Or increasingly environmental work. One last tidbit that's exciting to the graduate students that I hadn't appreciated. There is an absolute mind-blowing revolution underway in the renewable energy sector. And that's leading to the possibility of electricity generation abundance. Very recently, it's become possible to convert electricity into carbon by fixing carbon from the air and then using that to feed microorganisms. So you go from atmospheric CO2 to formate, as an example, and then modify the microbes to grow on formate instead of glucose. And so what that means, napkin math-wise, is you can take a kilowatt hour of electricity and turn that into a gram of new biomass. And if you talk to the material scientists and chemical engineers, they think they can up the efficiency maybe 20 to 30-fold. So a kilowatt hour of electricity gets you 20 to 30 grams of biomass. I don't think this is interesting in terms of reducing atmospheric carbon, but it's very interesting in terms of local manufacturing. And so this is an analysis that came out in PNAS, just imagining if you had a hectare of land and were growing soybeans, how many people could you feed a year? About 40. But if instead you had solar panels, made fixed carbon, and then fed microbes, and you had microbial protein production, you could feed about 10 times as many people. And I'm not trying to put this in competition with agriculture. Obviously, you could do this in places where you can't farm, right, for reasons. Okay, so now let me go way over and like when I put this together, what do I wish for or imagine? 50 years ago, there was a revolution in computing because computing became personal. It wasn't a revolution in transistor physics. It was a revolution in the terms and conditions by which people could access computing. So playing with that, imagine I have a box and in comes electricity. That's helping me fix carbon and nitrogen and other things. In comes biocode that gets printed into DNA, and the air is supplying the atoms. And what comes out of the box is whatever the box can make. And maybe personal is a step too far. Maybe it should be a family biomaker, or a village biomaker, or a house district biomaker. I'm not sure. But the basic idea is we're going bits to atoms and we're growing locally, right? And so that would mean instead of designed in California and made in China and globalization, we'd have a complement to that, deglobalized, distributed local biomanufacturing. Remember the opening of like, I'm freaking out about Mark Bloch and his book. And I'm worried that we're not tracking qualitative change correctly and imagining, what might be really new. And so we're being conventional in our biostrategic thinking and not at least hedging. If what I just said on the previous slide's right, another consequence of that could be anyone anywhere could be able to access any toxin or pathogen, right? And if that's a possibility, then maybe this is a good conservative starting assumption for thinking about biosecurity. How would you secure a biofuture in which anyone anywhere could access any toxin or pathogen? And is that different than how we're thinking about it today? David, you mentioned the iGEM. iGEM is 20 years old this year. I also briefed the DARPA director on synthetic biology 20 years ago this past Sunday. And so it's interesting to reflect on both those things. Normally you give a DARPA briefing as a nerd like me and you try and get money for a program. But my briefing was like the Hindenburg coming into New Jersey. I was talking about DNA synthesis and wanting to get better at DNA synthesis. And very naively, I was not correctly understanding the politics of the time post the Amerithrax attack And the DARPA director correctly looked at the naiver version, I'm still naive, but the even more naive version of me, said this guy just wants to make bioterrorism easier. And so instead of getting the program funded, we actually contributed to decisions that got all public funding in the United States for DNA synthesis pulled back. We got negative money from that briefing. Now, because of that, when we went back to MIT at the time, we were a little bit beaten down, but the people who wanted to work with us were the teenagers who were matriculating to MIT. It's like, it's the biocentury. Where's the bioengineering department? Somebody's going to have to teach me how to build life forms, right? Like if I was in electrical engineering, you'd teach me how to build a computer. Very reasonable set of expectations for a teenager, especially an audacious, ambitious teenager. We didn't know how to do that, but at least we knew that we could work together. And so that has become this thing known as the iGEM, the International Genetically Engineered Machine Competition, about 100,000 alumni. So it's a little bit bigger than Stanford now on an undergraduate annual basis. And the next one will be in a couple weeks in Paris, and I hope the bed bugs are all taken care of. But interesting, like Macron's underwriting this now. And I'm not sure if he'll be there or not, but we like endorsed it. Here's another thing to share related to iGEM. So this is an analysis. Is Connor here? Thank you. Thank you very much. So this shows participation by country or region over time in the iGEM. And so the blue line in the middle goes up and peaks around 2014 and comes back down. That's the United States of America. And there were very funny things that happened in the United States around 2011 to 2012 regarding synthetic biology. We had a little ice age politically. I'll take that offline. It's with the pandemic and synthetic RNA vaccines and DNA synthesis that synthetic biology is now back, right? But I learned about this from the AI folks, like in AI winter. Right? We had a little ice age in synthetic biology for the last decade. But check out the red line. Guess who didn't? Our colleagues in China. And so if this trend continues, by next year, the participation in the iGEM will be majority PRC. Okay. Now, how come of that? Here's something we did together through the Committee on Science, Technology, Law. We ran a joint trilateral effort with the British and the Chinese on synthetic biology, and we co-developed a roadmap for synthetic biology, and we successfully installed it in London and in Beijing. And as evidence of that, the Ministry of Science Technology came back to the United States in 2012 and gave us the roadmap back to us. The entire US delegation pulled out of this meeting 3 days before the meeting. We have no roadmap, right? We can't talk about this. But you look at their roadmap, it's pretty good. All the things you would imagine. Because I want to get to conversation, I'm not going to go deep into it, but I do just want to point out their 20-year target, which is 2032, because of the 2012 roadmap. Look at that last bottom right bullet point, creation of artificial microbial life. And at the time, somebody putting that on a roadmap in 2012 is really audacious. But if you loop back to what I was saying 7 minutes ago around our cryo-EM construction work, like I think we're about $100 million away from that, right? Which would be sooner than 2032 if you could organize the capital, right? But at least they'll get there by 2032. And by the way, if they get there, I want to go there as a nerd, because that's where bioengineering is going to be, right? And that's where all the PhD students are going to want to go, and so on and so forth, right? Like whoever gets that first, that's a big prize in biotech. Okay, so now let me try and pivot into policy and setting up some conversation. Here's from the State Department, March of 2022. State Department's responding to the Russian allegations of chemical and biological weapons labs in the Ukraine and refuting that. Like, nope, that's wrong. But check out the highlighted text. It is Russia that has active chemical and biological weapons programs and is in violation of the Chemical Weapons Convention and Biological Weapons Convention. One nation state accusing another nation state in public of an active bioweapons program. So how do I remix this sort of experience with my thinking about where biotech's going? And I'll give you an example that's a little bit childish, but maybe it's not totally childish. So I don't know, it's been a busy time. Maybe you didn't see the news reports. October of this month, the Russian Ministry of Health reported a new variant for SARS circulating in and around Moscow. And WHO confirmed that just a few days ago on the 7th. And the genome was finally sequenced and shared in the Western sequence repository. So everybody has the sequence. And just a couple days ago, the Chinese Academy of Sciences reported on the mutations unique to the variant. And suddenly multiple accounts on X or whatever Twitter used to be, right, is reporting that if you translate these triplets into amino acid code, you get a very interesting combination of letters related to Ukraine, right, in blue. And next day, I guess tomorrow, Russia is going to declare that they're under biological attack from a synthetic variant of SARS. And then intelligence from Russia is going to release a bunch of purchase orders from a US company, Twist Biosciences, that makes DNA, saying that gene fragments encoding these sequences were sent to the Institute of Virology in Lithuania. And the company in the United States later this week, next week, will deny it, right? We didn't do that. And they screen all their synthesis orders. But then additional intelligence says the Estonians hacked the American company, compromising their security measures, and inserted a sequence for synthesis downstream of their sequence checking, because the companies only check what's being synthesized on the front end. They don't check it on the back end after the DNA has been made before they ship it. And Twist says, yeah, you know, you're right, we don't do that. Oops. But we didn't do this, says Twist. And we certainly didn't send anything to Lithuania. And soon thereafter, there's accusations, right? More serious than this, perhaps. And this was serious, right? So what's interesting about this example for me is I could imagine this example happening, and it's all made up. It's just an information attack. The virus never exists in nature in the first place. The sequence has been added to the databases, purchase orders have all been faked, right? And it's just kind of stupid. But it's an example of a hypothetical vulnerability that if somebody was thinking seriously about abusing fake bio for geopolitical purposes, like you start, and put this in the context of all the other things we're struggling to navigate that I'll get to, right? So just, and just to be very clear, I've used this a couple times in classes, so I'm gonna say it out loud. This is all made up. This is not real. I don't wish for this to be real, but I've gotten emails after I've used this in 1 or 2 classes from students like, this has happened? Like, what? All right, okay. So let's just bring back to now questions I'm sure we've all confronted. Where did SARS come from? Have we implemented a system of governance, i.e. policies, that make it harder or easier to know where SARS came from or where biothreats come from? Have we made decisions that make it harder or easier to know? Are the things we're doing? Could we do better? Could we create, strengthen, or sustain trust in biology, biotrust? So I'm going to explore this a little bit more. The late Secretary Schultz had some very interesting things to say in this opinion piece he offered. In this context, I want to highlight these words. He wrote, "Trust is the coin of the realm. When trust was in the room, whatever the room, good things happened. When trust was not in the room, good things did not happen. Everything else is details." So I'm really curious about this idea of trust in biosecurity, in bioscience, and bioscience research. Thankfully, emerging biotechnologies are not the first emerging technology. There have been other examples of powerful emerging technologies. And the favorite one I've got for this topic of trust is called money. At a certain point in human history, money gets invented and emerges. And we quickly learn that money is a dual-use technology. Very dangerous. And it's very dangerous in part because I can't trust you. I can't trust any of you with money. And you can't trust me. Maybe. How would we know? Right? So how do we help money as an emerging technology to do good? Well, we develop a whole bunch of auxiliary technologies to help money. And one of those set of technologies is trust technologies. And when I say it in this way, it's very abstract and maybe even sounds sexy, but in practice, it's not that sexy. It's the financial audit, right? What is a financial audit? It's a private party-to-party confidential transaction that results in an inspection, and then that inspection produces a public trust certificate that can be used to represent to others whether over the most recent period of time, what you did and whether your practices are compatible with being trustworthy regarding issues and matters related to money. Right. Whoa, that's pretty good. Now bring this back to biosecurity and geopolitics and trust in biology. We don't have a lot of that. We have the collapse of inspections around the Bioweapons Convention because it all landed in nation-state industrial espionage, right? So could I get PwC and the other accounting firms to create new bio-audit divisions? Would that be good or bad? Okay, let me set up one more policy puzzle / question. David mentioned I'm on the Variola committee for WHO. We're meeting later this month. There's no funding for that committee, so we're meeting by Zoom. That'll be at 3 a.m. in the morning Zoom. And smallpox is horrific. There's no other way to say it. But the good news is it's been eradicated through a vaccine campaign, at least as a public health issue today. But if you're not familiar with smallpox, it's just hard to fathom. In the last 100 years of its existence, of order 500 million people died, right? At a point in time where the average human population is 2 billion or something like that. And it's eradicated through vaccination and the public health efforts of many. Here's an incredible statue at the entrance to the World Health Organization in Geneva and the vaccine campaign, somebody getting a vaccine. On the top right is another incredible artifact. This is the smallpox eradication program poster from Ethiopia. Right? Like, what does it look like to operate a public health campaign on the ground and get people to uptake vaccines? And compare that to how did we do with COVID vaccination, right? So with this as backdrop, the interesting thing about smallpox in today's conversation for me is how we govern research with smallpox. And David, this goes back and is meant as a bookend to your very important remarks here the last time we were talking about matters related to this red line. So if you were to ask me, can you do research with live smallpox as a member of this committee, the default answer is going to be no. That's it. It's very different than how we approach research with influenza and coronavirus and other potential pandemic pathogens. The answer is no. Now you can start to make an argument, however, if we need to do the work in order to get a better diagnostic or better treatment or better vaccine, and only those reasons, and not some fancy scientific question, but if we need to do the work to get a better diagnostic, treatment, or vaccine, and we need a better diagnostic, treatment, or vaccine, then maybe. But you have to propose the work to us, and a multilateral committee will review it, and we'll probably say no. Unless you can convince us that you really need to do the work and the world needs you to do the work, and the work requires working with the live virus and not some other thing in order to get a better diagnostic, treatment, or vaccine. Right? So a default no governance regime for research with a virus. Now, to be careful, smallpox is different operationally than flu and coronavirus because the human reservoir for smallpox is not complemented by animal reservoirs, whereas for flu and corona, right, you have animal reservoirs. But there are things like monkeypox, right, and Alaska pox. Thankfully, not so much. But it's interesting. Like, did you know we have implemented a default no multilateral governance regime for working with a pathogen? What would it be like if we adopted that governance regime for other pathogens? Would that make establishing trust easier or harder? I think easier. So here's my last slide just to summarize and tee up policy puzzles and ideas. I view that ambiguity, residual or imagined, that remains regarding COVID origins, that needs to be viewed as a policy failure because of the choices we've made around how research is conducted and resourced. And I haven't heard these words stated too often publicly, but I view the resulting ambiguity, and especially in our broader society, it's a policy failure. The internet represents, combined with DNA read-write, a third regime for biotech. And it's very different than what came before. And it's still emerging, which means it's like if you were in 1970, you were talking about internet governance, what would you do? Or social media, what would you do? What would you want to get right for network security if you could go back to 1970 and do over, right? So that's where we are right now with third-gen biosecurity, if you will, in my view. Starting assumptions regarding biosecurity should be revisited. Like, what if anybody anywhere could get any pathogen or toxin? Where is control best implemented in a bits to atoms world? Is control reasonable? Is it possible, or is it harmful? From a different perspective, recall we're governing a press, a printing press for DNA. So how should we govern a press in a way that's compatible with a flourishing democracy, right? And citizenship within a democracy. Here's something that I think we need to debate and perhaps bring back to the biomedical community. The arguments against default no governance of research with pathogens is scientific knowledge is paramount. If we don't advance scientific knowledge, we're in trouble, right? If the pathogen mutates or something like that. But I think what that misses is political trust. So could it be the case that political trust is more strategically important than scientific knowledge for certain types of work with pathogens? And in my view, the answer is it is more important. It's increasingly more important for things like coronavirus and flu. We should go default no on those. So that's just restated here. Again, noting the caveats, these are circulating things with animal reservoirs, and so there is a background cost to further restrictions on the research. Lastly, I tried to get this across, but it wasn't the main theme. The physics of flourishing is really good. If you're tracking what's going on in renewable energy, and you can see biology getting unlocked as a general purpose technology, if we could responsibly deploy that, you can get to multilateral flourishing. Not at some arbitrary point in the future, but just as soon as we get on with it. And so what that means is, rather than only frame things through the lens of bilateral competition, right, or limited competition or zero-sum competition, I think there's a whole other side of this strategy is how do you lead around multilateral flourishing? Like, what's our Indonesia strategy? Would be compiling that down to a more actionable question, right? They're going to be the fourth largest economy in a couple decades. They've got massive biodiversity, huge carbon reserve, really interesting supply chain puzzles. Like, how do we partner with Indonesia to get to flourishing, right? So thank you very much. And I'm glad we have some time for questions and discussion.
Drew, first of all, thank you. And I too am delighted that we have time for discussion. As you all are quite familiar, we will start with students, fellows, trainees, and then move on to those of us with greater numbers of years. And although I guess all of us are students in many ways. And the one finger, two finger arrangement as well. So let me first ask whether we have questions from the trainee cohorts. Yeah, and if you could just let everyone know out in the ether who you are before you ask your question or make a comment.
Right, does this work? Can you hear me? All right, brilliant. Hi, I'm Max. I'm one of the CSAC fellows working on AI safety. And thank you for the great talk and your insights into this topic, because I have a different background, so it was a lot of new information. Thank you very much. And I was wondering, could you please help me better understand the jump where we currently are with the technology and where we might be? Because I understood the example of how to create the artificial life with the Slack project. We're seeing, okay, we're getting there. But you also mentioned the case in an initial slide, if we would have the better technology where a PhD student is currently working on with the lasers and you do the ups and downs, what can we currently synthesize? So we can do viruses? But what will be able...
the limits of DNA synthesis and construction, viruses are all within reach. Horsepox was demonstrated in 2016, regrettably. So that's of order 200 kilobases. Right, so any viral genome less than 200 kilobases is within striking distance for a team that has the tacit knowledge associated with the process of booting up that particular virus. Right, it'd be much harder to do if you were, like a kid, right, without that implicit knowledge. But that's addressable over time. Then beyond that, most notably, There have been projects around genome minimization and genome synthesis for microbes, right? So entire bacterial genomes, rewriting native genomes have been realized, 500 kilobases. And then there's a consortium organized by Jeff Buka and others called Yeast 2.0. And so they've been rebuilding the chromosomes of Saccharomyces cerevisiae, which is about 12 megabases in total. And I believe they're done, but I don't think the celebratory paper has come out yet. That's a eukaryotic organism, right? So synthesis has gotten to the point of eukaryotic chromosomes. Right, so about 10 megabases. The synthesis process itself is limited to short fragments max, right? And then there's stitching and assembly that goes up to tens and hundreds of kilobases and megabases. But those stitching and assembly processes are really neat. They're done in biology, right? So yeast is a DNA assembler once you get up to those length scales.
Drew, can I just add to that question my own,
Yep.
which is that when I think about your third epoch, this synthesis era, I still see largely a focus on design that has already been provided to us. And in some ways, the absence of the kind of imagination and rethink of what a design might look like or could look like, other than those for which we have ample examples already. And I'm just wondering whether we've focused too much in thinking about our progress on the nuts and bolts of making rather than the much harder task of imagining design and how design might be.
I completely agree with you, right? So just anecdotally, a good colleague and very creative geneticist, George Church, we were both at a meeting, and I said, George is working on recoding E. coli and other amazing things. And I said to him in private, but I'll share it here, like, George, how come you're so boring? Why are you just recoding E. coli? Like, why do you just want to bring back the mammoth, right? Is another project he's working on, right? It's like, why so boring, right? And that got him to smile, right? Because I think you're right. It's very hard to escape the constraint of lineage. I want to give one technical example and then zoom back out. So here's some work we published 4 years ago now, and we were redesigning the genetic code itself such that all mutations might be deleterious. So the natural genetic code evolved in nature is evolved in a context of a changing environment, and it's fairly good at enabling mutations to be neutral or maybe not totally harmful, such that some might be beneficial. But imagine you're a rocket engineer and you want to be able to responsibly launch a rocket. And if the rocket's going off course, you want a fail-safe that blows up the rocket, right? So now instead of a rocket trajectory, imagine an evolutionary trajectory through lineage space. And if the organism mutates, you want to be able to select against it, right? So that it stays on the nominal path. For that to be true, every point mutation would have to be deleterious. Now, such a genetic code in nature, if the environment's changing, would it go extinct, even if it randomly occurred? But in synthetic biology, because we don't have that constraint, you just make it. We started prototyping that and showed in theory that there is such a thing. And it's like, right? But even that's still pretty close, right? So, one of the other hats I wear is over at the D School, right? And the co-director of the degree programs over at the D School, right? The design school at Stanford. And the D School starts in mechanical engineering and product design, but in its next generation, it's adopting different domains. And one domain we're adopting is called living matter, and how to bring designers into the world of living matter to imagine what we might make with this stuff. I don't think the creativity is going to come from me, right? Like, I'm not creative in that way. These folks are unbelievably good in imagining new things in a way that I'm not.
Yeah, and I guess one answer to this is, well, you start with function, and
Yeah.
then you work back through known structures that mediate those functions, but you're still constrained by the structures that we already know to emulate.
And this is what I was trying to get at. It's like this metaphor of being up on the lip of the life well and looking around and what could it all be. The other thing that's going to be, coming back to your introduction to yourself, interesting is how generative software is going to land in this space, right? So it's starting already with proteins and AI hallucinating proteins. But if we can get the physics engines right, which I now think we can at the cellular scale for cellular scale systems biology, then you're going to see generative software start to suggest or just hallucinate things at the single cell level, right? And then it just keeps going. So that's another wrinkle in all of this. I'm not, well, we'll see, have to see. Yeah.
Harold, I saw you had your hand up.
Just, I really want to return to another metaphor you used in your talk, which is of the personal computer.
Yeah.
And the connection to the internet and how transformative that was. And so as we know, in terms of, your concern about having like your own personal sort of synthesis device or family synthesis device I guess your analogy was that the PC sort of brought a lot of good, but also created certain harms, right? Like viruses and ransomware and hacking. And so in terms of imagining responses for these highly distributed kind of potential sources of harms, there's the model that looks like, I don't know what model makes most sense to you. Is it that highly distributed kind of antivirus, like constantly checking every day to make sure you're secured against the latest thing? Or does it look more like the way the internet deals with spam, which is systems operators are, there's the statistic from, it's probably over a decade now, that 90% of email is spam, but 90% of your email is not spam. And so that's being done at the system operator level, sort of out of our sight, not on our desktop, it's being done out on the internet. And so in terms of this connection, atoms to bits to atoms, which is kind of what we see in the cyber world, can we imagine is there something there that you would pull on, or is there even valid analogy?
I think you're right on, Harold, and it's restating it. For me, understanding approaches to network security and network strategy are like, that's where we got to go. We're just hosting the leads of security for Google in a conversation with a couple colleagues in computer science earlier this week, and listening to how they thought about platform security, network security. An example comment was, nobody's checking code at the compile step, right? And in this world, it'd be nobody's checking DNA at the build step, right? I'm like, well, how do you deal with it? And the answer is provenance mostly. Like, we are very carefully tracking where the compiled stuff came from, where the stuff came from, right? And so that was interesting, and I have to think about that more. Another thing they admitted to was patch hell, right? Like, it just all the time, right? Vigilance and patching. I guess what I'd like to represent is, and maybe this ends in no good result, but it feels to me, like we may be too early on this one. How does network security strategy apply to future biosecurity? We may be too early on that. I don't think so. But I think we're definitely not too late yet. The other thing I should say is, what's interesting about synthetic biology is there's nobody like me who has unfettered access to money yet. And this is important for two reasons, right? So think about AI. There are a lot of unilateralists who are nerds in the realm of AI, and they have plenty of cash. And so it's always hard to explain what you think the next neat idea might be. Like, no matter how good you are at communicating, it's just hard, right? And like convincing somebody to give you limited treasure to do something. But once the nerds have unlimited cash or enough cash, you get this different flywheel spun up, right? And like, everybody like me right now is still begging for money. And that's a disadvantage in terms of geopolitical competition, because you want to let the nerds loose at some point if you're really racing. But it's a wonderful privilege in terms of having a little bit of time to think about it, right? So when I was at OECD back in June, talking about synthetic biology, I was trying to give them a temporal runway. It's like, you have about 3 to 4 years to, don't think about how you want to govern this by reacting to it. You have 3 to 4 years to express your version of the social contract, what must be true, what should be true, what could be true, and what won't be true, and express how that should manifest in terms of the next generation of biotechnology. Like, now's your time to do that. And they're like, huh? Normally we're reacting to Sam Altman, you know, because he's like futuring all over us with the latest ChatGPT patch, right? And so it's this very interesting window. And coming back to your question, you're exactly right in my book. It's like network security approaches. I also wanted to give an acknowledgement, right, also on the nuclear and physics side, right? So in response to the abstract for today's talk, Chris Chiba sent a nice note of a paper he wrote looking at the history of recombinant DNA governance, but also nuclear governance in the 1930s, and how Szilard was working to stop or delay publication of certain things and ultimately failed, right? And so there's interesting lessons there around control of information, and if you try and lock things down in certain ways, you know, where does it squeeze out? But network security feels like a big deal to me. It feels like the big deal to me. Like, we need a network security strategy for all of this. And I don't know what that means beyond putting those words together right now. Yeah.
Yeah, in the back of the room.
My name is Leo P. Sahin. I'm a political scientist at NYU. I'm just visiting here as a scholar. Very interesting talk. I had 2 quick questions. So to continue this conversation about the future of this and how to secure this, I wanted to ask you specifically about China. So in your personal opinion, what should that look like? Is it a cooperation where, you know, people working in the US or in Western Europe should just say no to cooperation with China because there is no trust? Or should it be a trust and verify system where you cooperate until you find that something has gone wrong and then you stop cooperating? Or do you only cooperate in some domains and not others? So that's the sort of policy question. And then on the science of this, which I know nothing about it, I just find it all very fascinating. You know, if this synthetic biology sounds like a solution to scarcity, it's like, you know, if this really works in 40, 50 years time, we will have solved the problem of scarcity, right? That's what it looks like. Is that actually true? Or is that a bit utopian? So what potentially, is there going to be a new scarcity that results from this method? Are we going to be, is a particular type of material, chemical, something else going to become scarce because this is going to be the dominant technology? And if so, what is going to be that material or whatever it is that will become scarce?
Yeah, so let me take those questions in reverse order. Thank you for visiting and joining today. It's a great set of questions. I don't have a full audit on the supply chains around getting to abundance, but if I were to give you an example, and I'm learning, I'm a student of all of this. All right, let's take photovoltaic solar. Right, the last year I have data, our civilization built a terawatt of panels for photovoltaic generation. Right, and in doing that, best I can figure, we use 10% of the silver supply. Right, if I'm tracking this correctly. And again, anybody else could easily be a better expert on this than I am. Right, but I'm trying to backfill because I have the same operational questions you do. Historically, and there's not that much history here, panel manufacturing capacity is doubled every 4 years. Right, so if we did a terawatt a year ago, right, then in 26 we're doing 2 terawatts of panels and 20% of the world's silver, unless mining changes. And then by the time we get to 2030, we're doing 4 terawatts of panels a year, and we're eating up 40% of the silver supply. Right, so it's like, I'm not willing to, like, even that's gonna be tricky, but let's just play, we can go that far. That's kind of interesting to me, if something else doesn't become limiting, which it could, and I just don't know about it yet, because we're doing 4 terawatts a year of panels. That's every 5 years, we're stamping out 20 terawatts of panels. That's a civilization worth of energy every 5 years. We've never encountered that before, right? And the reason I started going down this path is, like, initially 10 years ago, I put 10 solar panels on my garage roof, and I think about it as a consumer. You know, what does it cost me? What's the electricity worth? When do I get my money back? What's the ROI? But I started thinking about more as a physicist. How much energy does it take to make the panels? And how much energy does the panels make? And when do I get my energy back? Return on joules or return on energy. And now you can find really interesting analysis in the sector for the whole sector of manufacturing panels. In 2010, the ROE for photovoltaic is less than 1. You don't get your energy back. And so physically, it only makes sense to put them in places where you don't have better options, like a satellite, right? But sometime in the 2010s, we went above 1 as a sector. And by 2020, it's 20 to 1. You get your joules back in a year. Return on energy is faster in return on money, right? And that's never been true for renewable electricity source before, in my understanding, that I can see. It's why Stanford, we installed or rent 1 million solar panels now in 2 energy generation stations in the Central Valley that offsets the entire campus electricity load, right? Like we did it, right? And we could keep going if we wanted to. So I don't have a full audit, and I would love for a group to go at the abundance audit seriously, holistically. I don't think of it as a utopia. I think of it as a protopia, progress, right? Now let's come back to, I still got to get to your China question, but you said your background's, your political theory. Political science, great. So nobody's figured out how to navigate the politics of abundance, to my knowledge. We seem to depend on regimes of scarcity to maintain political stability. Sometimes the scarcity is true scarcity, and sometimes it's artificial, right? And so it's like you're navigating through the rural roads of New England. It's like, you can't get there from here. Like, how do we get to flourishing, right? And in a political science sense, nobody solved that puzzle. You know, from Marx to Marcuse, you correctly diagnose the ills, but nobody gets the escape hatch open, right? So just want to flag that, and would really value deep follow-up on how to navigate that transition constructively, you know, not in some disruptive revolutionary sense necessarily. China, I think you started part of my response in your question, where do we cooperate, where do we compete, where's there conflict? I'll give you an example related to today. Let's take at face value that maybe there are countries that have offensive bioweapons programs, right? I gave one public example. Well, could we convince them to shut it down? Like the United States, we shut ours down under Nixon, right? Using logic. And so if you had an authoritarian regime, such as China, maybe you could make the argument that you developing a bioweapons program, maybe your motivation is to offset power, but you already have plenty of conventional power and nuclear power. So then the other reason to do it is to suppress domestically and maintain power domestically. But if you could successfully convince such a country that ultimately you're not going to be able to control the bioweapons because anybody can get them, and they ultimately are a dire threat to your authority, that might be able to tip them over and get them on board to be an ally around turning off bioweapons, right? And I've only thought about this twice, including today, just now, right? But so like maybe we want to cooperate on that. You know, and I mean that as a short-term strategy. On a longer-term strategy, I have different thoughts, right? I think biology is democracy's natural ally, because it's the ultimate distributed manufacturing platform. It provides optionality with respect to access to essential capacities. And so ultimately, over the long time scale, I'm naively optimistic in a way that seems eerily similar to how the free marketeers thought free markets would get to free people. And I might be at risk of wanting to rerun that playbook, but around manufacturing. So again, like a placeholder for needing help and following up. Right now, if I come back to the iGEM, my view on the iGEM is it's an incredible platform for coordination, and the values of the iGEM defined by the rules of the competition and the core technology stack, which is defined by the United States right now. There's a lot of importance and value in that, a lot of soft power in that. Even if the competition becomes majority PRC, so long as the values, right, remain and the tech stack are ours, to be frank about it, it's a good. If that splits or forks or goes sideways.
Do you have a two-finger and then Scott?
Two-finger questions regarding China and the nourishment for all. In the AI comparison, even though we have a lot of proliferation and open source models and everything, as soon as you have really powerful AI nowadays on the future, you have a sphere of a concentration of power. And do you think the proliferation with these kind of technologies will be large enough to outweigh a similar fear where just one country, like actually China, another country, would just have these really strong biotech capabilities without the weapon component, just for building more energy, building more food, and the nice bridges? Do you think it will work out in that case, or is it something to worry about? Thank you.
I think there's going to be network systems result in coordination solutions, right? So like, where are you going to go for search? you're gonna go to Mountain View, right? Google, right? And that's an entrenched coordination solution. No matter what Musk does to what used to be Twitter, people are still using Twitter, right? Because that's what you use, because it's the entrenched coordination solution, right? So as biology becomes ever more networked, the opportunity to create coordination solutions, like where do you go for the code base, the DNA code base, or where do you go for the expertise around industrial translation, or where do you go for the biosecurity strategy, or where do you go for the creative design work? You know, like where are those hubs going to be that get locked in? And once they get locked in, right? So I'll give you an easier to understand example. The #1 law school in the United States of America before the rankings all went away was in New Haven, Connecticut at Yale. And that's because their coordination solution to a coordination problem that college undergraduates have, which is where should I go to law school, right? And I want to go to law school with the best law students. And the problem is, the coordination problem is you don't know who the other best law students are. You can't get them all on a Zoom. And even if you could, you couldn't get all of them to agree to go to New Haven, right? But because Yale has entrenched itself as the place to be, that's the coordination solution to the coordination problem. And if we're #2 here at Stanford, it's almost impossible to displace Yale, right? And you don't do it by fancy faculty and fancy buildings. You've got to do sustained attack on the decision-making process of the undergraduates, right? And good luck with that, right? So I do foresee that there are going to be these winner-take-all, on the positive side, things that get established in the biospace, which adds a type of urgency for me. Normally, when you think about emerging technology, we think of it, for example, through the lens and experience of Moore's law, like a tools making better tools, tech exponential. It's like you got to keep up, because if your competitor races ahead, they're advantaged and accelerate away from you, right? And so we're familiar with that type of competition, right? But in a network strategy domain, because there's the risk of entrenchment, you have this extra urgency, this double urgency of you need to get somewhere first, right? And I'm not saying it perfectly well, but I do think, you know, it's like we see in the economic sphere, let's just play it out this way. Imagine the Earth is less and less easy to live on for humans, and people become more and more desperate for solutions. And biology has something to offer in terms of a suite of solutions, right? Whoever has those solutions at the ready will have tremendous geopolitical power if they're willing to share them. That's another way of thinking about it. Maybe that's easier to think about. Scott.
Yeah, Scott Sagan, I'm also a political scientist, and I loved your use of fail-safe as a metaphor. You're learning something from Cold War nuclear things. But I like less well your comments about George Shultz and political trust, because although Shultz often would talk about the importance of trust, his president said trust but verify. You want to have some trust, but you also want to verify because because trust is not enough. And certainly in the area of smallpox, there's good reason to do that. The most recent claims about Russia and China give you to do this. So I'd like to talk a little bit more about verification of activities, what's possible. And here I think the metaphor for fail-safe might be what was called the NITSE criteria. When people said, well, you can't catch all cheating, and you can't have all verification, everything can't be verified. He said, yeah, you don't need to verify everything. You just have to verify just enough that the person doesn't have a massive advantage. And therefore, let's get off of this idea about perfect verification. Is there something in that analogy that could be helpful in the biosecurity space?
I think so, but I'm sure I can't think of all the ways. So first, thank you for that comment. There's a lot in that, and I agree with the extension of the time. So, what can I do to make it easier to do verification, right? So let's say you didn't trust me, and I wanted to make it easier for you to verify what I was doing. There's a whole bunch of things I could do to make it easier. So for example, I could publish an encrypted hash or similar around every piece of DNA I ever make. Now you'd have to believe me that I was doing that, but imagine I was doing that and that was world readable, and you could inspect that, and by inspecting it, determine if I made anything that you were concerned about or not concerned about, right? So I don't know if you want to pull in zero knowledge proofs or those sorts of ideas, but the basic idea is, what might we do to enable as many actors as possible to make it easier to do verification by getting as much as possible out into the open where it can be inspected more easily. I think there's a significant amount of work that can be done there. It doesn't address, a fully clandestine nation state actor that's going off the grid and doing work, right? But think about all the ambiguity around origins of SARS and how much better we could have done if we just had as a standard of practice, all researchers were leaving behind a trail of everything they were doing that was inspectable, right? And so just as one part of a response that's not sufficient to your comment, I think we could do a lot with that idea of starting by making verification easier. But it's so interesting right now how closed up things are, right? So it's hard to do worse in terms of verification, I think. Yeah.
Herb.
A two-finger on Scott's question there. I mean, what Drew has been talking about is the equivalent of on-site inspection in some sense. The NITSE criteria was, whether or not they would verify it to a level where they wouldn't gain a substantial military advantage from it. And the problem in biology is that just a little bit of stuff of anything is bad. I mean, Drew's into this totally is self-replicating. Or is potentially self-replicating, and now you have a prospect where something small that's small today can turn into something really big tomorrow, and that is much more significant. So it's much harder to implement the NITSE criteria under the circumstances of biology, where missiles don't self-replicate, whereas viruses do. I think that would be the concern.
Comment. Milana.
Thank you so much, Drew. I love learning from you. That's why I keep coming to your talks. Although I've heard a number of them now, but I feel like they're always different. I learn every time. To echo a little bit, some of the sentiments that were discussed. So verification is tough, and like I've heard from the Lawrence, actually the head of the safety of Lawrence Livermore Laboratory, that one of their best strategies is actually kind of boils down to the culture of trust, kind of to make it a funny circle back to trust, and George Schultz's comment, because it is so difficult. Somebody could, put a little, walk out with a little vial of material straight out of the lab, and you would never know. That's in this country, and other countries, you can only imagine. So of course, as Drew, you were talking about, with the proliferation of technology, it democratizes things, right? But with this democratization of the technologies and of all the amazing things they can bring, it also democratizes risks. And as an expert, and I know we've touched on that, with the democratization of risks, there's also increased fear, right, of these technologies. So from your perspective, and I know I've asked you that before, but maybe, you had additional kind of new thoughts. It's so hard to control it in a centralized way. So what can we do really to make this abundance safer? That's something I struggle with. I'll be honest. I don't have an answer to this, despite how many times I come to your talks.
Yeah. So, thank you for the question, Milana. I return to a call to examine starting assumptions related to biosafety and biosecurity, because I'm suspicious that we're holding dear historical practices related to and starting with physical containment of pathogens that are increasingly outmoded. And so an example, which I don't wish for, but put forward, is what if anybody anywhere could access any pathogen or toxin as a conservative, worst case scenario operational starting assumption for the future of biosafety and biosecurity? that turns then into a positive challenge. How do we make that not matter, right? If that's a possibility. I don't know of an example where a technology gets secured without, especially a distributed technology that's not limited by access to fissile material, right? But it's a technology, everybody's got biology, without people having understanding or the option of having understanding of the technology at the periphery, right? We're coming up on the 50th anniversary of the very famous recombinant DNA safety meeting that Paul Berg and colleagues organized at Asilomar in 1975. And at the time, gene editing had just been invented, and there were ideas about, what was it, cloning a oncogene into a bacteria for studying it. And if that got out and people ingested it, would you get cancer, right? And so this very important meeting happens. It happens in the blessing of the standing down of the offensive program in the United States. And so they're primarily tackling the topic of biosafety, setting aside biosecurity. They're confronted with the topic of fear of biotechnology in general, and the scientists are not equipped to navigate fear as a topic. So they run away from fear. They have fear of fear, right? And so they put those two things together, the safety concerns and the public fear of biotech, and they move genetic engineering into the biosafety laboratory framework that takes this emerging technology, implicitly acknowledges that it's scary to the broader public and hides it away in laboratories, right? Because of that, if you go back to the Dutch social scientist Huizinga in the 1930s and his book Homo Ludens, Man as Player, I come to the conclusion that we don't have a culture yet of biotechnology, right? And so from Huizinga to Donna Haraway, you find examples of puppies learning how to do dangerous things like bite each other. But they do that through play. Like, I'm gonna give you a bite, but it's not a real bite. I'm using this dangerous thing, my powerful jaw and sharp teeth, but just a nip. And when you stop and think about everything behind that experience, there's communication, trustworthiness, honesty, right? So many things get established. Also understanding of limits. So many things get established through this seemingly simple behavior of play. And so Huizinga argues that play has to happen before you can have a culture that establishes norms and other practices. And we've never played with biotechnology. And it's always been locked up. And because of that, we don't have a broader culture of biotechnology. So here's an example of a type of play. This is actually genetic engineering work that was done in Russia and then been licensed by a US company called Light Bio. And they just got approval from the US government to sell these plants. These plants are modified to have a cassette from a forest mushroom that glows in the dark, that emits light. And so by adding that genetic cassette into petunias, you have petunias that grow on light, but in the dark, they emit photons. They're night lights. When I brought some of these prototype plants home for our boys still in elementary school, these were the first things that they thought were cool. Previously, they only thought of GMOs as evil and bad, right? Because that's what they learned in the public school. And that's what they see in the Jurassic Park movie, where the evil corporation is called Biosyn, a literal transposition of syn bio, right? And so like the only thing that moved the needle for the boys on public personal acceptance of biotech was nightlights that they water, as opposed to plug in or change the battery on. And we just don't have very much of that. And I think, by the way, the government approval for this is wonderful, but kind of also not impressive. There's almost no consideration of ecological impacts that I can see around insects or other behaviors. Really impressively oblivious. Anyway.
So, Drew, I just wanted to maybe bring this back to one of your first conclusions and ask you to maybe elaborate. For those of you who aren't aware, I think we're at a really interesting moment when policymakers, at least in this country, but also others I know, are thinking a lot harder about whether there are tangible steps that could be taken now to try to anticipate and move a little bit faster towards oversight that makes sense for technologies that are moving so quickly that there's a little bit of fear in their minds, perhaps. The White House just issued a request for ideas and response to suggestions they're making about how to strengthen an oversight regime of this work on pathogens that could lead to a potential new pandemic pathogen. So you concluded that the problem with our failure to understand COVID origins was a policy failure. In your response to their request for information next week, what would be the first couple things that you might suggest to them in the way of policy that could matter, that might go beyond a call for greater cultural sensitivity and norms and so forth, and get a little more edgy?
Well, my penultimate one here, right, I'm willing to push on and would like more debate and dialogue on. I think we should default to no for other pathogens like smallpox. If Professor Luby was here, I won't speak for him, but I'd be willing to bet he might agree. And so I'd like to push on that one. The point just above, I think, is not directly policy, but is important for policy making. Historically, as I've experienced it around governance of this type of research, the most important factor has been ignorance kills. We need to protect unfettered science so that we can figure stuff out. And there's valid reasoning behind that, but it's very one-dimensional in a more complicated landscape. And so I was, in prepping the notes for today, trying to do this, like maybe political trust is more important than scientific knowledge. Like, when is that true? And get that type of thinking into the policy development process, because right now it seems very one-dimensional and myopically focused on discovery, right, at all costs, right? The third thing I would put forward is, I don't think you govern DNA synthesis by checking what people are making through a centralized screening process. And I'll note our local representative, Representative Eshu, recently advanced a bill, not sure where it is right now, introduced a bill, right? That starts off by saying we should develop next generation approaches to governing this, but then the remainder of the bill says what the answer is. And it's the answer I helped come up with in 2005, right? Which is a good idea back then, but maybe we do better. So I just want to flag, I think you're grateful you're pointing out this is a live topic in terms of executive branch policy and legislation. So I'll just repeat and conclude my answer. Maybe we should default no for other pathogens. When are other factors more important than scientific knowledge? And how do we get the apples to oranges to potatoes balancing right? Because right now we're just looking at the apple and go, we need to eat the apple, right? And we've got to figure out a way of governing this capacity to go from bits to atoms while being mindful of freedom of the press, democracy, citizenship, and other stuff. And it's all too reactive right now. Yeah.
Drew, I want to be mindful of time. It's 5 o'clock exactly. I know we're leaving a question or two here on the table, but Drew will stick around. And for that, and for a wonderful talk, I just want to thank you immensely. And please join me.